From the moment of fertilization, a single African clawed frog oocyte transforms into 10,000 individual cells in under 10 hours. This rate of expansion represents an extreme instance of biological acceleration, where a single cell must partition itself into a multicellular organism at maximum kinetic velocity. To understand the speed of this process, we can compare it to the division rate of a standard mammalian somatic cell. A typical mammalian cell requires roughly 24 hours just to complete a single division cycle. This sequence shows the early morphological milestones of the Xenopus embryo. Following an initial 90-minute setup, the zygote enters a phase of synchronous cleavages that occur every 25 to 30 minutes. Maintaining this precision means that by the 7-hour mark, the embryo has already reached the 5,000-cell mid-blastula stage. This speed creates a mechanical conflict. Cell division is typically rate-limited by the time required to accumulate mass and the need for quality control checkpoints. To achieve a 30-minute cycle, the embryo bypasses the standard eukaryotic failsafes that usually prevent a cell from dividing before its DNA is verified. To reach the 10,000 cell mark on schedule, the embryo discards complex regulation in favor of a minimal, biophysically optimized oscillator powered by maternal materials. A standard somatic cell cycle consists of 4 distinct phases: G1, S, G2, and M. In normal cells, the G1 and G2 gap phases are used for cellular growth and as checkpoints to ensure the genome is error-free before the cell proceeds to mitosis. The Xenopus embryo eliminates the G1 and G2 phases from its early cleavage cycle entirely. Growth is unnecessary because the massive 1.2 millimeter egg was preloaded during oogenesis with the proteins and mRNAs required to reach the 10,000 cell stage. The embryo even keeps its own genome silenced during these divisions to avoid the metabolic delays of transcription. Stripped of these growth phases, the system is driven by a biophysical engine called the maturation promoting factor, or MPF, complex. This circuit operates via a bistable regulatory loop that depends strictly on the concentrations of CDK1 and cyclin B. This oscillator autonomously toggles the cell strictly between DNA synthesis and mitosis, ignoring the growth-based inputs used by somatic cells. Simplifying the cycle to this raw 2-state oscillator provides the structural framework required to maintain the 30-minute division interval. This 30-minute cycle creates a biochemical bottleneck during the DNA replication phase. The embryo has a mere 15 minutes to replicate its entire 3.1 billion base pair genome. Because the speed of DNA polymerase enzymes is kinetically limited and cannot be increased, the embryo must find another way to move faster. The solution is a massive increase in the density of replication origins, the starting points where DNA synthesis begins. While somatic cells space these origins far apart, the embryonic genome activates them every 10 to 15 kilobases, allowing thousands of segments to be copied simultaneously. This speed has a genetic cost. Without checkpoints, the embryo uses translesion synthesis polymerases to jump over DNA damage sites, which prevents replication stalls but increases the overall mutation rate. Prioritizing speed over genetic fidelity is an evolutionary trade-off for an organism transitioning from a vulnerable egg to a motile tadpole as quickly as possible. Beyond replication, the embryo is limited by the physical scale of the egg itself. Molecular diffusion is too slow to synchronize biochemical events across the 1.2 millimeter volume of the egg. It would take roughly 2 hours for a regulatory protein to diffuse from the center to the surface, far longer than the 30-minute cycle it needs. The embryo bypasses this using self-regenerating trigger waves of CDK1 activity moving at 60 micrometers per minute. These self-sustaining trigger waves utilize a localized positive feedback loop, propagating outward much like a forest fire rather than drifting randomly through the cytoplasm. Local MPF activation triggers neighboring activation, ensuring the signal reaches the edge of the massive egg with no loss in speed or amplitude. These biochemical waves visibly manifest on the embryo surface as ripples of contraction and relaxation moving across the cortex. These trigger waves allow the embryo to achieve flawless spatial coordination across thousands of cells, despite its enormous size. Eventually, this rapid cleavage must stop, so the embryo can begin the process of cell differentiation. This phase is defined by reductive cleavage, where the total volume of the embryo stays the same, while internal cell count doubles every 30 minutes. The embryo detects when to end this phase by monitoring its nucleocytoplasmic ratio, the balance between total DNA and cytoplasmic volume. Without a temporal clock, the embryo uses a chemical titration mechanism to physically count its divisions. This count is tracked through maternal histones. The egg begins with a fixed pool of free histones that actively suppress the activation of the embryo's own genes. As the cells divide, the total mass of DNA exponentially doubles every 30 minutes. This increasing mass of DNA acts as a sponge, soaking up the free histones from the cytoplasm to package the new genetic material into chromatin. In the early divisions, the concentration of free histones remains high enough to maintain transcriptional repression. When the free histone pool finally falls below a critical threshold, the biochemical blockade is lifted, marking the onset of the mid-blastula transition. This transition brings the rapid cleavage to a halt. The cell cycle slows down, asynchrony begins, and the G1 and G2 gap phases return. With the repressive histones depleted, the embryo's own genome finally activates, synthesizing the messenger RNAs needed to form the 3 germ layers. By stripping away regulation and relying on maternal biophysics, the Xenopus embryo achieves its 10,000 cell milestone, using its own replicating DNA as the brake that transitions it into a vertebrate.